Okay, welcome back after lunch. Good afternoon, everyone. This is state-of-the-art lecture number 5 and the last one. It's a pleasure today to have Dr. James McKay from IAC, who will present on genetics and cancer. So Dr. McKay is the deputy head of the genomic epidemiology branch. He has been working in genetics and genomics for almost 20 years and on a number of different cancer types, including lung, head and neck, kidney cancer, and also lymphomas. James was trained in Australia. He's from Tasmania originally, and then did his PhD there, but spent time at IAC, then went back and then eventually rejoined IAC a few years back. So he's going to give some example on how genetics is used within IAC research. Thank you, James. The floor is yours.
Thank you very much, Pietro. I've had the good fortune of knowing Pietro for many, many years. And when I was a student, way back in the day, we had some great skiing adventures together. And it's a real pleasure to be introduced by him today. I really wanted to start out first to thank every one of you. Like, this is supposed to be an expert, but I had a fantastic meeting with some of you. I can't see them. Yes, I can. From Bangladesh and Malawi yesterday. And I felt that interaction at the back there too, I thought that interaction was absolutely wonderful. And I can tell you, well, if I'm supposed to be the expert, you guys in this room inspire me as much as the other way around. So I really appreciate you being here and interacting with us. And I hope to give you some ideas about how we use genetics here at the agency. So what I'm going to try and do, it's supposed to be a state-of-the-art, so some of it will be quite technical, but I've also tried to keep it as simple as possible, knowing that the background in the room is quite varied. And so what I hope to do is explain some of the research that we do here at the agency, and also explain why we do that. Why would we want to use genetics in this particular way? And what I try to do is give some lessons that I've done in that context across the work that we've done in lung cancer. As Pietro said, I work in other forms of cancer and genetic susceptibility in other forms of cancer, but these lessons apply to all these different forms. So here's a great diagram. This is what I'd start about. What does the agency actually do? I think, as Pietro said, I'm number 5, so I think you've heard a lot about the other aspects of the agency and what we do. So where does genetics fit into this? It really is coming in. We're not necessarily describing the occurrence. The guys in the CSU unit are doing a great job of describing the occurrence and how cancer changes across different countries and across the world. But what genetics actually is really involved in is more the panel on the left over here, understanding the causes. We can use genetics to describe that. And secondly, more and more, as we understand genetics more and more, we can be more involved in implementation and prevention. So what do I mean by that? Why do we use genomic epidemiology at IAC? Well, because genetics can do a number of different things. Firstly, if we can understand genetic profiles, we can identify people at risk of developing a given disease. It doesn't necessarily have to be cancer. It could be many different types of disease. And then we can try and manage that risk clinically once we've identified those high-risk individuals. That could be through therapeutic interventions. It could be through improved diagnostics, improved surveillance, and so forth. So if we can identify those high-risk individuals, we can manage that and make sure that clinical risk is managed appropriately. So that's a real potential that we can do, and I can think in a couple of examples. Particularly, there's genes called BRCA1 and BRCA2, which markedly increase the risk of developing breast cancer, and I'll come back to that a little bit later. But the agency, while we're very focused on this aspect, we also look at it for another couple of reasons, and these are perhaps equally important for the work that we do here at the agency. First of all, we can understand the biology. Now, a lot of the techniques that I'm going to describe today are called agnostic genetic analysis. And what this means is we don't think about a particular hypothesis. What we try and do is look at all the genetic variants in the given genome. And that way, we identify novel aspects. So genes that we wouldn't have thought a priori would be involved in these particular diseases. So we can make these novel findings. Through those novel findings, we can understand a little bit better the disease itself, how it develops, what the pathways are, and therefore better understanding about how we can prevent it and develop novel biomarkers. There is also a lot of activity looking at new novel therapeutic targets, so that's not necessarily the focus of our work here at the agency. But there's another equally important and interesting way of using genetics here at the agency, and that's to look at the gene-environment interactions. So the idea behind this is if we can identify the genes that are involved and then discover that they've been modified by the environment, that can give us some clues about how the environment works to influence cancer susceptibility and so forth. I've got a great example of that, and that's in head and neck cancer. In the Monographs program, which you may have heard of, the Monographs program was involved in classifying whether or not alcohol was a carcinogen. Is alcohol a carcinogen for humans? And at the time, some of our genetics work, the fact that the germline genetic variants modified that process, was used as part of the evidence to suggest that alcohol was a carcinogen. So we can use that idea. But the other way we can do that is also look at novel environmental exposures and use the genetics to try and reverse engineer which environmental exposures are involved in the disease. And I'm going to give you an example in a second or two about how we would do that in the context of lung cancer. So this is the two aspects that we try and do here in genetics. We're really focused on understanding the causes of cancer, why that comes together, what are the biological the mechanisms involved in cancer development, what can we learn from genetics from do that, and then also by using the risk profiles to try and identify those who risk purpose. So that's how we use genetics and genomics at the agency. And there's a few other aspects, but they're the core areas that we try and do. Now, I just wanted to go back a little bit because when one talks about genetics, one needs to make sure that we understand what we're talking about. So most of the germline genetics that I'm going to talk about today are those constitutional DNA changes that you've inherited from your father and your mother. Overall, we have 47 different DNA molecules, the autosomes, which are the 22 chromosomes that we've heard, the sex chromosomes, chromosome X and Y, and the mitochondria. Now, each of these have different aspects to them, and the genetics changes a little bit. One also wouldn't forget the bottom part, epigenetics. That's also something we inherit, and it also is involved in a number of different types of diseases, although it's less well studied. Most of the things that I'm going to talk about today, though, are really focused on that top part, the DNA molecules that we inherit from our parents, particularly the autosomes. Now, what about human genetic diversity? These things, what is interesting in this is that we do actually vary across the genome. And this can be a number of different ways. There is an aspect that I'm going to focus on later for most of the talk, but there are multiple different ways the genome can vary. The first thing we can actually look at is how the chromosomes are actually different. For example, there's a particular example of a pericentric inversion where chromosome 8 has actually been inversed in a number of individuals. So the whole chromosome of chromosome 8 is fundamentally different between different individuals because part of it is actually twisted around and placed the other way. The functional consequences are quite minor, but nonetheless, it's a major change between individuals. There's other parts of the DNA which are a lot more, much more focused in their change. And on this screen here, you can see a repeat here. You see in the DNA stretch that I put up on the strand here, you can see that repeat of CA, CA, CA. And if we, in many parts of the genome, when we look at these types of stretch, this might vary between individuals. So here there's about 10 different CAs, but if we looked across different individuals, that may change, where the repeat may be 8 or 6, 12, and each individual has inherited a different number from their parents. There are other types of this type of thing where there's chromosome heteromorphism, mini-satellites, microsatellites, and this can actually even apply to entire genes, where you would have different numbers of copies of a particular gene across different individuals. For example, one person may have one copy of that gene, others may have 2 or 3, and that again influences the amount of expression that happens. But the one I'm going to really focus on today is this bottom one, and these are much more subtle changes where there's only a single base pair change in the DNA. So on the bottom of the screen here, we can see that that red base pair has been changed from a C to a T. So that's only a minor change in the overall coding sequence, but nonetheless, if it's in the wrong spot, that can actually have an important consequence. And this may be one of the most important contributions of overall genetic diversity in the human genome. This can also be in other forms of this subtle change. For example, in the bottom right here, you can see that the sequence GTGCT has been where there's 2 bases that have actually been deleted. So the actual 2 bases have actually gone. And again, that can impact on the gene function. So with that in mind, what are the consequences? What do these changes actually do? Now, this is something that's really important to remember. Most of the genetic variants that we have in our genome actually have no effect on any gene product. So most of them fall into intragenic regions or in between the genes themselves and don't actually affect at all the functioning of the genes around them. And so most of these things that you've inherited from your parents will actually have no impact on the phenotype or your well-being or your manifestation of your human nature. However, some do. And then perhaps this is, while it's less than 1%, these are the ones we're probably most interested in. And the nature of the change is really also quite varied. Some variants actually do have quite a major effect on the gene product. So we call these nonsense mutations. This type of mutation will actually result in the loss of function of a given gene. So this will actually cut the gene in two or something like that. The net result of that, the gene doesn't work anymore. We also have missense variants. So this is a change in the amino acid structure. This can influence the impact, the protein's function once it's transcribed. There's also gross deletions, and this results in the complete loss of a given gene itself. And again, that has quite an important function, impact on the function of the gene itself. Remembering, for the autosomes at least, we usually have 2 copies. So there's also the recessive diseases and the autosomal dominant diseases. So if we lose 1 copy, it's not necessarily a problem because we have a backup copy in a way from the other parent. But again, that aspect is some of the nuances of what we do inside genetics. And there's another type of variant that have a lot more, much more subtle effects. So these, and this is what I'm going to focus on most of my talk today. Originally, when I did my PhD with Pietro 20 years ago, I really thought the majority of the genetic variants that influence cancer susceptibility would be these nonsense types of mutations, the things that have quite an impact on the gene products. But in fact, it's actually things that actually have much more subtle effects. For example, they change the expression levels. And I've got an example of this one here. So up here, we've got 3 genotypes. So this is a common genetic variant. We have, of course, 2 copies. We have this particular individual has 2 copies of TT, and in the middle, there are 130 people with the heterozygotes. So they have 1 copy of the alternate allele and the second copy of the wild type allele. And there's another copy where they have 2 copies of the alternate allele. And on the y-axis, what we see is the expression levels of a given gene. Now, this particular example, what's happening is if you have a copy of that wild type allele, the net effect of that is that increases the amount of expression of that gene. And this is in proportion to the number of alleles that you have. If you have 2 copies, you get twice as much expression, or increased expression relative to the wild type copy. And this type of effect seems to be one of the major drivers of cancer susceptibility and phenotypic variants in humans, where we see this influence on gene expression. And the effect is quite subtle. This particular gene, it's involved, we're not exactly sure what it involves, but nonetheless, people vary. And in this particular gene, we have associated that with increased susceptibility to lung cancer. And I'll explain a little bit further a bit. Now, what do we expect about the consequence of these genetic variants? As a general rule, this graph really explains how the phenotypic impact on cancer susceptibility. So on the y-axis, what this is plotting is showing the effect of the alleles on cancer susceptibility, so the genetic effect. If you receive one of those alleles, what's the consequence or the relative risk of developing that disease over time? And on the x-axis at the bottom here, that is looking at the population frequency of these particular genetic variants. And what we can see is this kind of inverse association, where the rarer those alleles are, the more striking the effect. And on the y-axis, you can see that, in the bottom right, that as they become more common, the effect on the phenotype, for example, cancer susceptibility, becomes much more less pronounced. And we can actually, whoops, just gone too far. So up in the top left, this would be like the gene variants that are involved in susceptibility to breast cancer, BRCA1 and BRCA2. If you inherit those genes, you have an important effect on, important risk of developing breast cancer over your lifetime. And the green dot in the middle, that would be more something we call moderate risk genes. So these genes are genes, for example, ATM and CHEK2, they increase over maybe 2 or 3-fold your risk of developing a given cancer type over your lifetime. And then in the bottom right there, these genetic variants are much more common in the population. And when I mean common in the population, within this room, for example, we would expect between 30 and 50% of the people to actually have these susceptibility alleles. Now that's not alarming because the magnitude of the risk is actually still very modest. So while we have an increased risk and we can demonstrate statistically this is related to risk, the impact on you as an individual is still quite low. So as in the context of clinical genetics, we would not be thinking that this is a meaningful risk in terms of developing that cancer over the person's lifetime. It doesn't mean we can't learn a lot about it. The other thing that's important to remember, on the bottom axis here, we could also replace that with the degree of function. The rare variants that seem to impact the phenotype quite strikingly tend to be the ones that have a major impact on the gene function, whereas on the ones over here that are much more common in the population, they're the ones that have a more subtle effect and tend to be the ones that influence gene expression. And for the rest of the talk, I think I'll focus on those ones that are common in the population. The other thing to do is, why would I bother talking about it? Why would we be interested in looking at different types of genetic diseases? So one of the first things one does as a geneticist is actually establish that the trait we're interested in is actually involved in genetics. Many traits, there's nothing to do with there is nothing to do with a given genetic trait. One can argue that if I stepped outside and was hit by a bus, that had nothing to do with genetics. Perhaps you could say there was some risk-taking behavior or something like that could be contributed by genetics, but many things, it's nothing to do with genetics. And one way we can actually estimate the contribution of genetics of various different diseases is look at twins or other ways of measuring heritability in a given family. And this example that's on the screen here, this is looking at the risk of developing a given type of family when you have a family history of it. So a first-degree relative that's related to the cancer. What's the risk if you have a first-degree relative that has a brain cancer? What's the risk for me as an individual of developing that same type of tumor? And what we can see here that there is elevated risks in a number of different cancer types. So you can see here that breast, colorectal, bladder cancer, it's about between 1.5 and 2-fold, 2.5-fold for lung cancer, prostate cancer. And then in some types of cancer, this seems to be quite marked. For example, lip cancer, chronic CLL, chronic lymphocytic leukemias, Hodgkin's disease, another form of lymphoma. These genetic risks are really quite high. Now, lung cancer is a really interesting one because one of the other things that within lung cancer, as you probably all know, one of the major risk factors of lung cancer is tobacco smoke. This is one of the biggest risk factors for developing, and it's a very important risk factor. And the other thing we also know, in a given family, you are more likely to smoke if your family smokes. And that makes a lot of sense because if you are primed as a child to tobacco smoke through passive smoking, you're much more likely to take up that habit later in life. And that would influence this estimate, because if we're thinking that these cancers are genetic, well, we should see many more in the first-degree relatives. But if the environmental factors influencing the fact that the cancer's clusters inside the family, we would see excess inside the first-degree relatives, not due to genetics, but because of that shared environment. And as an interesting example of this, here what we've done is we've estimated this heritability using the germline genetics themselves. And what we see is a slightly different story, where we still see quite remarkable increase in heritability or the geneticsness of this trait in prostate, in breast cancer, but lung, it's much more modest. It's still significant, but it's much more modest. And that we take to mean that this is the influence of that environment in those observational studies that looked at twins and so forth. So that was the net effect of that was the heritability was overestimated when we looked at the families, and it's a bit better estimated when we look directly as using the genotypes. Let's skip over that. Oop. Okay, so how are we actually studying it? How are we actually here at the agency trying to understand the heritability? And this is the method of choice that we've been using. There's something called a genome-wide association study, or a GWAS. You've probably heard this word, a GWAS. What is it? It's actually really, really simple. We take a bunch of cases, so patients with the disease, and a bunch of controls that don't have it, or at least haven't manifested at that time. We then simply test each genetic variant, just test simply which are they more frequent in cases compared to controls. It's that simple. The only complexity is that we have these wonderful chips. This is a genome-wide association screen. This can actually assay almost all the common genetic variants in our genome, and there are a lot of them, about 7 million of them. And that's where the complexity of these things start to happen, because when we do these tests, we're actually not just testing one genetic variant, which is a very simple thing to do. We could do that on a bit of paper. What we are testing is 7 million genetic variants between these cases and controls. And if, as any good statistician will tell you, if you do 7 million genetic tests, everything weird and wonderful that could happen to those in that particular thing will be observed in that particular experiment, because we've done the same thing 7 million times. So we need to be very, very careful about how we assess the statistical significance of anything that we find. And there's a bunch of different rigors that we do when we perform these different studies to try and control and make sure that our studies are robust. But the one thing that's great is that we're very, very accurate in measuring these genotypes. So therefore, we can actually make some given the right settings, some quite interesting insights, and these results tend to be robust when things go well. And this is an example about how it works. So this is a map of the human genome that I took last year when I was preparing a given talk. And this demonstrates each one of these little dots has one particular phenotype that's been related to a genetic disease using this approach, this GWAS approach. And there are almost 30,000 now genetic variants that have been implicated in various different forms of disease. And of those, there are about 3,000 that have been related to susceptibility to various forms of cancer. So this particular approach has been really, really successful in understanding or increasing our understanding of germline genetic variants and how they influence human traits. In the context of lung cancer, we've been doing this since 2008, since our original findings in 2008. And just last year, we published this study, and this gives you an idea of the scope that we're trying to do. So our last study involved about 61,000 cases of lung cancers and almost 900,000 controls. And it's one of the most interesting aspects of the agency is that we're able to coordinate these different types of studies, because to assemble this type of thing, we need to be able to assemble studies from all over the world to get this type of sample sizes and work with researchers all over the world to try and put it together and then analyze this type of data with all the challenges that go with it. So these are the genes that we've identified so far, and there's been many of them. And many of these may make sense to you. So things about, for example, DNA repair, that makes a lot of sense to someone who's smoking. If you're smoking a bunch of tobacco smoke into your lungs, if you have a genetic difference about how you can repair that DNA, that would make sense. That would subsequently result in an increased cancer risk over your time. There's other genetic variants that are involved in telomere length. This is the edge of the chromosomes, how long they are. That seems to play quite an important role in susceptibility to lung cancer. There's other things that are involved in somatic biology, for example, NRG1, which is a really interesting gene that was discovered by a colleague of mine here at the IARC, and many others that are involved in things that we don't know. And that's one of the aspects is really interesting for this type of work, is that we're able to identify things that we have no idea what they do. But because of the process that we do, we can establish that they are causally related to this disease, and that then the onus is on us to actually explain it. For example, that gene, the first one there, which is awful to say, SECISBP2L, I really wish we should change that. I propose that we name it Pietro. It's much easier to say. Would you like a gene named after you? No, yeah, it'd be a good gene. It'd be a good gene. That's the one I gave you the example of how it modifies the gene expression. So the susceptibility allele changes the gene expression levels and in turn somehow modulates lung cancer risk. How? I don't know, but I know it does. And so now the onus is on us to then understand how that works and what that can teach us in the biology. Now, the other thing that I'm going to spend most of the time about, and this is a really fascinating story, is that the genes that are, many genetic variants seem to be related to propensity to smoke. And that makes an awful lot of sense to a disease like lung cancer. We know that the observational studies have shown us that tobacco smoking increases your risk of lung cancer. So if you have differences to the degree which you smoke, driven by genetics, that should in turn modulate your risk of lung cancer. And they're the stories I'm going to tell you about now. The other thing that we were very, very lucky, I'm going to spend a couple of minutes discussing this, about how we have integrated a bunch of different data sets to understand the biological processes. Because the community at large, like I showed in that sense, there's been 30,000 genetic variants across the genome which have been identified. So there are hundreds and hundreds, if not thousands, of traits that have been analysed. And so what we do is a bunch of computational biology and computational science to integrate different layers of genetic evidence. In this instance, what we're trying to do is look at the intermediate trait. Do these influence smoking variants? Are they in telomere length? Is it related to gene expression? Or is it related to metabolism? All these different type of things we try and integrate into our studies to understand the genetic variants a bit better. So how do we do this? Now, I'm not going to spend too much time on this, but it's really worth in place. So the idea behind this is something called co-localization. And this is I'll see how I go if I can explain this clearly or not. Sometimes it works, sometimes it doesn't. One of the attributes I think I mentioned is that in the human genome, there are 7 million genetic variants. And then we have to think, well, of them, how many of them are associated with a given trait? Actually, quite a few. This is a particular genetic area of the chromosome 22 that I've plotted here. And something we can note, there are 6,000 genetic variants across this particular thing but only 11 of them are associated with lung cancer. Okay, now if we were thinking that this might be mediated through a gene expression or something like that the genetic association is coming through the change in a gene expression or something like those, we can assume those same 7 variants must be also associated with the genetic trait. If it was a separate set of 7 variants that have nothing to do with the ones that are related to susceptibility, that would imply that it's not that gene. Okay, it's that simple. So if we find a few genetic variants that are associated with lung cancer, they should also be associated with the second trait. And we can use that concept to examine if that trait is involved or not. Equally, if it's not, we can exclude that hypothesis. And now I'm going to give you an example about that. So here's a great example. This is a genetic variant. I think it's on chromosome 8. It's related. We were able to establish that it is related to lung cancer. And we also noticed that it was related to gene expression of this gene called RNA-seq2. We see a lovely dose response where if you have one allele, you increase your expression a little bit. 2 alleles, it increases a little bit more. And so what we do is we do this type of plot, which is something related to co-localization. There's a statistical framework to do this, but I always find it's really great to plot it because you can visually see what I'm talking about. So here, there's 7,000 variants at this particular point in the genome. What I'm doing on both axes, I'm just plotting the amount of association. In this particular statistic, it's focused around 0. If there's no association at this particular locus, it should be all centered at 0. And of those 7,000 variants, that's exactly what we see. With a bit of variance, all of them are all not associated with either trait. But however, we actually see, because I'm plotting on one axis the association with lung cancer, on the bottom axis the association with gene expression levels, we see a correlation. The alleles that are associated with lung cancer seem to also be the alleles that are associated with gene expression. This doesn't look like chance. So therefore we can say, well, we've just generated the hypothesis that this association is mediated through changes in gene expression. There are many variants associated with neither. Some variants that are associated with increased expression and increased risk, and some variants that are associated with lower expression and decreased lung cancer risk. It seems to fit. Can make it a little bit more beautiful, put a bit of color in here and add an aspect of linkage, something called linkage disequilibrium or LD. And this is to look at how correlated each genetic variant is, and probably want to go into that here. Okay, let's look at another example. Same thing. There's an interesting genetic variant. This one's on chromosome 11. It's robustly associated with lung cancer and also is involved with gene expression levels. But But if I do that same plot, we see a much more complicated situation. There doesn't seem to be a clear association or clear correlation between the genetic variants associated with lung cancer and the genetic variants associated with the expression levels of this particular thing. The key variant, the one that I've noted here, RS105, that's a way of expressing a particular genetic variant, that is associated with both lung cancer and gene expression levels. Increased risk of lung cancer, decreased gene expression. However, at the top there, the ones in sort of green color, one can see clearly that those ones are associated with lung cancer that seem to have no impact on the gene expression levels. And then we've got another group, which are the ones in green on your far left. Those ones, they seem to be associated with gene expression levels, but not at all associated with lung cancer. So there's this really inconsistent relationship between the alleles that are associated with gene expression and the ones associated with lung cancer. So we think it's less likely that this particular, the modulation of this association is driven through gene expressions levels of this particular gene. So we can perhaps put that candidate aside. So I'm going to give you an example now about how we use this idea. So this was one of the first genetic loci that we associated with lung cancer, and it contains some really interesting genes, particularly these acetylcholine receptor genes. Why are they interesting? Well, they're involved in how the human body modulates the effect of nicotine. They are involved, they're neurotransmitters, so they have a natural effect in our body. But when you smoke a cigarette, as nicotine circulates through your body and particularly in your brain, it's through these particular receptors that the effect is actually modified. And there's a couple of different ways that individuals vary between that these particular receptors vary between individuals. For example, there are missense variants in these individuals. So some people respond stronger to nicotine than others. There's also gene expression differences. Some have more expression of these receptors than not. If we look at the lung cancer evidence, there's clear evidence with lung cancer. We can see across many different things. So what I'm doing is this is something called a forest plot where I've plotted out many different strata of lung cancer. If you're a smoker, how much do you smoke? Does it differ by how much you smoke over your lifetime, in the number of pack years you smoked over your lifetime? Does it differ by age, or does it differ by gender? And what we do see is that in some aspects it does seem to differ. You can see here that there's very little effect, for example, in never smokers. This would be logical if the lung cancer that's presenting in a given individual is unrelated to smoking. The fact that you've got a nicotine receptor probably means it should be null in never smokers. And it would appear so that it is. It's not, the association is only through smoking. And the other thing that's really important in terms of evidence is we looked to see how much each of these different allele carriers smoke per day. You can see here on the far left, this is looking at the three genotypes, the homozygote, heterozygote, homozygote rare. And what I'm doing is just plotting the number of cigarettes you smoke per day if you started smoking cigarettes per day by genotype. And as you can see, the risk allele, the one that increases the risk of developing lung cancer, is also associated with the amount that you smoke per day. So if you have the risk allele, you tend to smoke a little bit more. And that makes a lot of sense. One, we can think that if you smoke more, then you're going to have an increased carcinogen exposure. Over your lifetime, you're going to increase your lung cancer. And that seems to be the way that works. You can do the same trick as I showed you just previously with the gene expression levels. We can plot this. So this is this particular locus on chromosome 15, and we can see a lovely correlation between the genetic variants that are associated with lung cancer and the ones that are associated with smoking more per day. So we can have a bit of confidence to think that this association appears to be mediated through modulation of how much you smoke per day. Now, we found this really interesting locus on chromosome 8. And here there were two really interesting genes. There was one of these genes that are involved in nicotine, the alpha-2 receptor. Okay, that's a really interesting gene. But there's also this gene called APX2. And this is also equally interesting because that's involved in how people metabolize tobacco smoke. And maybe this is about how, if you metabolize a little bit better the carcinogens in tobacco smoke, that would mean you don't get as much carcinogen exposure. and therefore less risk over your lifetime of lung cancer. So both of these are really great candidates, but what's the evidence of them mediating this association? What could we use this notion to look at which one's the better candidate? And that's what we did. So the first one we did was looked at that one EPHX2. This is the one that's involved metabolization. And we saw a really inconsistent relationship. The variant there, I've noticed on the right here, RS625042887. It's a very long name, but that one's really associated with the gene expression levels of this gene, but it's not associated with lung cancer. Go ahead, please.
I have a question. So these variations are normal variations or consequence of somewhat of genetical mutations?
These are constitutional DNA. So these are normal things that one would find in anyone's genome in this particular building. They're not somatic changes that are happening through your lifetime. Does that answer your question?
Yes, yes.
Thank you. So here, what we're saying here is that this particular individual, if we take the room, we genotype every person in this room, there are certain people that will have higher expressions levels of this particular gene, and certain people will have lower. The net effect of that, in this instance, it doesn't change the risk of lung cancer in this particular example. And this gene, you can see a very big difference between this plot and this plot. Same genetic variants, but in this instance, while this candidate gene, EPHX2, seems to be not correlated, if we look at the other candidate, which is this nicotine receptor, it is correlated. And so what we're trying to think here is that this particular gene is more likely to be involved than not. So this prompted us to then go and look at this particular genetic variant and see if this was related to smoking propensity. And that's exactly what we found. I'm going to zip through this one. And here, this is this particular genetic variant. One curious thing, it didn't change the amount you smoked. It seemed like it was different. That first genetic variant, it changes the amount you smoke once you have started smoking. This one, for whatever reason, tended to result in you taking up smoking. You tend to be a smoker rather than a never smoker. You tend to take up smoking a little earlier in your life, on average, than someone who doesn't have this genetic variant. The net effect of that is the same. You end up smoking more over your lifetime compared to the others. Net effect of that, more carcinogen exposure, more genetic risk. Now, exactly how that works, we're not sure, because there was something a little bit strange with this. Usually addiction is something cerebral. It's the top of your brain. And this particular genetic variant seemed to be very much involved in the cerebellum, the bottom part of the brain. And this was a bit of a surprise. And we were able to replicate that in 2 different data sets. This genetic variant only changes expression in the cerebellum. And all the folk that were involved in addiction were really surprised at this particular finding, because usually addiction, tobacco smoking, is something that happens in the cerebellum, at the top of your brain. Whereas this, it's something to do with the cerebellum, which is usually involved in motor function. And so one hypothesis that come for this, and I know people are testing this, is maybe this is related to motor function. That because the nicotine receptors just makes you, as you smoke your nicotine, it makes you a little bit more stable in terms of your motor function. And that could prompt you to take up smoking a little bit better, or a little bit more and earlier. Okay, there are many other genetic loci that we looked at. This is a really fascinating one in the CYP2A6 locus. This one governs how fast you metabolize nicotine. There's a really good example of this. I was just wondering how many people in this room had a coffee at lunch today? Did anyone have a coffee at lunch today? The Italians did. Okay, there's a really interesting gene that governs how fast people metabolize caffeine. If you have a genetic variant, you metabolize caffeine a lot quicker. The net effect of that is you will drink 30% more coffee per day than someone that doesn't have that. So I would guess the people that put up their hands probably have that genetic variant. Now, just to reassure yourselves that put up your hands, there's no evidence to suggest that changes in any way your risk of any diseases. But nonetheless, it modulates your behavior, because to get the same dose as I'll add of caffeine, you have to drink more coffee. And I'll put my hand up. I have that allele. I drink a lot of coffee. This gene, which is a separate gene, it's very similar. This changes how fast you metabolize nicotine. So if you're a smoker and you have 2 cigarettes, a given individual that has this genetic variant, you have to smoke the same a little bit more to have the same net effect of nicotine dose per day. And we see again a lovely correlation between the genetic variants that are related to how much you smoke and the ones that are associated with lung cancer at this particular locus. Oh, there's a couple others. Now, the last thing I'm going to talk about today is actually this notion. If we were to look at our genetic variants and we think that they are related to how much you smoke, that's the hypothesis, that we would say that if you're going to smoke a little bit more per over your lifetime, this would result in the net effect of that. Your overall dose of smoking is a little bit higher than another individual. What we should see is the overall accumulation of somatic mutations. Now, this comes back to your question a little bit. Now I'm talking about the mutations that happen over your lifetime. So as you're breathing in tobacco smoke into your lungs, what's the consequence? If you smoke a little bit more, a priori would expect to see a little bit more somatic mutations in your somatic tissues because of that increased tobacco exposure. And we can have a look at this because, as we know, there are often patterns of mutations that happen that are related to particular carcinogen exposures. For example, if we were to look at people that have been exposed heavily to UV light, the net effect of that is you will see a very particular DNA base pair change related to that exposure. And for tobacco smoke, we actually do see a high propensity of this C to A change. So if we see in a given tumor a higher amount of those C to A changes, we can make some assumptions that that's being driven by tobacco smoke. And if we go back to our original idea, if we were to think that germline susceptibility is involved with that, we should see a difference by genotype in the numbers of somatic mutations in those tumors. So those with a smoking propensity should have proportionally more smoking-related changes in their somatic tissues. And we can test that by a process called polygenic risk scores, and that's what I'm just going to go through now. And so what we did is we generated a combination of scores, something called a polygenic risk score, and then we applied that to a bunch of lung cancer cases where the tumors had been sequenced. So we could quantify how many smoking-related mutations there were in each individual tumor. And the number was really quite large, almost 1,200 lung cancer tumors that we were able to study. And this is what we saw. So on the bottom axis here, we see the polygenic risk score. So this is how addicted you are. How much lung cancer susceptibility do you have along this axis? And on the y-axis, it's how many mutations that we see inside the tumors. So those people that were more addicted to tobacco smoke, they do have more mutations in their tumors. In fact, about 30% more. On the ones that don't seem to be addicted to lung, that have subsequently they actually have developed lung cancer, they proportionally have lower amounts of mutations. So this was again something that was consistent with this hypothesis that you are smoking more per day, the net effect of that increasing your carcinogen exposure, the net effect of that increasing the number of somatic mutations that you're accumulating over time, net effect of that increasing your risk of developing a lung cancer over your lifetime. We can do it this way. We can also look at that particular somatic mutation that's related to tobacco smoking, and we see the same thing. Where we see the polygenic risk score, it's wonderfully correlated with the number of mutations that you see inside the tumors. And this means that we can use genetics to try and explore what that carcinogenic process is, and that's really why we're doing it here at the agency. Again, if we want to be, we need to be also very conscious of chance. So what we're able to do is replicate that finding in an independent series to make sure it's robust. And that's paramount to all the different work that we do here at the agency. Once we identify a given risk factor or something like that, we try and make sure it's robust by looking at additional samples. Okay, I'm going to stop in a second, I promise. Here are the key points. So one of the wonderful things about genetics is that we can study diseases from an agnostic point of view. We can make these discoveries when we don't know what we're actually looking for. And by doing that, we can push ourselves in unknown and unexpected directions. There's heaps of other big data opportunities using all these different data sets to explore the findings that we actually have. And so what we're able to do is use the genetic studies of many, many different traits to try and look at the etiology of these different types of diseases. And there's many, many opportunities for clever ideas about how to do that. This approach has identified many, many different genes, DNA repair. I've really focused on the ones related to nicotine, but each of these has their own separate story. And we could discuss these others if there's interest. And the other thing is we've done is the lung cancer GWAS has really highlighted the importance of smoking. Now that may not necessarily be a big issue, but it shows a real proof of principle about how we can use genetics in this type of thing to explore the etiology of a given disease. And I'm just going to stop now to just acknowledge everybody that's helped me, because this is certainly not me that's done this. There's an entire consortium of people that have helped me over the years to develop this research. And I'm also very grateful for all the funders that have helped do this work. And I'm happy to take any questions if there are any. Okay. And I'm going to give you all a test now to make sure you're listening.
Okay. Thank you, James. That was really clear. Any question from the audience? By the way, that paper that you and I co-authored in, what was it? I think it was 2012 on alcohol.
I would like to just reaffirm to everybody that was the best part of my work. 2012, mate, I don't remember. Which one?
2012 was ADH and LDH genes and alcohol intake and colorectal cancer.
Sure.
It was probably the first study conducted in among Europeans, because these genes are very prevalent among Asians,
Yep.
but a lot less among Caucasians. And so that was back then we didn't know whether it was actually it was not a Mendel randomization study. It was a gene environment interaction.
It was.
That's right. And I was checking recently. That paper has been cited 52 times.
Really?
Yeah.
That's cool.
Yeah.
So this is, again, the hypothesis we're exploring there is if we looked at those genetic variants that are related to how, so those ones, they modulate how much alcohol you drink. Now, if we would think that through, if you change how much alcohol you drink, what would happen in the context of your alcohol, of your cancer risk, if that's causally related to alcohol consumption? So if we think that alcohol is related to colorectal cancer, what we should see, and this is the process of something that Pietro alluded to, is something called Mendelian randomization. And so if we can demonstrate through your genetics that you should drink more, the net effect of that, those people who drink more, if there's a relationship between alcohol and a cancer risk, they should develop over their lifetimes and on average more cancer. And if we see that, we can really sort of establish that that particular agent, in this instance alcohol, is causally related to that given trait. There's an entire another branch of genetics and statistical analysis that I know Pietro's branch is very heavily involved in, ours too, about using this approach to try and look at the causal relationship using genetics, because there's also some study design advantages in doing that. Mendelian randomization. Please.
Thank you for the lecture. Thanks for your lecture. I have another question about reversibility of genetic variations or mutations. Is it possible, or something like that, when genetic variation, genomic variation or mutation occurs, that stays there? For example, in case of lung cancer, we say if you quit smoking for the past 10 to 20 years, you are like somebody that have never smoked. Is the genetic background, if the smoking causes any mutation or any variation, it stays there and makes this individual more susceptible to develop lung cancer in the future?
Absolutely. That's a really interesting question. I mean, the notion that if we can, excuse me. Sorry about that. That was a really interesting question. The notion that we can actually reverse these things. Constitutional DNA, it is now possible that we can use something called CRISPR to edit someone's genome and reverse the possibility of having a genetic disease. There's a big ethical debate about that, but there could be some good clinical reasons why one would want to do that, because there are some very devastating genetic diseases that if we can identify in an embryo and intervene, that could really change the outcome of that particular individual. Now, there's an entire ethical debate about whether or not we should do that. So that's if we're going to change our constitutional DNA. In the context of these particular genetic variants, I don't think that's something we would do at this point. But there are some, and I know there's some clever ways of doing that in the somatic cells as well. So for example, if we would try to, we know a genetic disease really involves blood cells. We could, rather than trying to change the constitutional DNA for every single cell in our body, we could target that change to a particular cell type where the disease is relevant. It's another thing. Although at the moment, this is still science fiction. I know there's some interest in that, but demonstratably, it's not something we can do right now. And there's some big dangers, because where do we stop? Rather than thinking about disease susceptibility, maybe we can change it to be performance enhancing, or other aspects where we could edit our genome. And there's some real ethical rabbit holes that and ethical dilemmas one would need to consider before we start doing that type of disease. In the context of lung cancer, are those things reversible? If you start smoking at 20, and you quit smoking at 30, your risk of developing lung cancer, or even the mortality associated with multiple aspects of tobacco smoke, drops down. So it's pretty much that if you stop smoking by 30, the risk over your lifetime of dying from related to tobacco smoke is almost back to never smoking levels. Slightly different if you stop at 40, slightly different again if you stop at 50. The golden rule, stop smoking, that's that simple. And there is a risk reduction overall. The consequence of that, and does that remove the somatic mutations that have happened when you're in the 20s? There is some evidence at this point to suggest that, although we don't know, because it's actually very difficult to look at the histologically normal material and see what the mutations have actually happened. When we look at tumors, they're usually a clonal expansion of one particular cell type. But if we look at the normal cells, there are hundreds, thousands, billions of cells. So what's the consequence to an individual cell? Very difficult to do. And one of the most exciting research opportunities that we have now is that technology is now becoming so advanced that we can look at individual cells or groups of cells and try and look at the trajectory. And to address exactly that question, I submitted a grant, I'm hoping to hear in the next month or so, to look at long-term former smokers. What do they look like in the context of the histologically normal material? Have they lower amounts of tobacco-related mutations in the histologically normal material compared to other people that have never smoked, or compared to current smokers? What is that trajectory? And what can that teach us about risk of developing cancer? Because unfortunately, while there's so many benefits in stopping smoking, because of the large proportion of individuals that have smoked, lung cancer in long-term former smokers is still quite a big problem in terms of presentation. So we need to understand what are the additional factors that provoke that disease. And that's what we're trying to do in that grant application. It's a really good question.
Thank you for the lecture. I just want to ask a question about benign tumors. Do you study them and their ability to become malignant, for example?
Benign tumors. Yeah, that is actually a really, really topical one. I focused here on lung cancer, but at the start, as Pietro said, I work on a number of other tumors. One of them is lymphomas. And for example, CLL, chronic lymphocytic leukemias. It's a really interesting disease because it is a very indolent nature. There is a benign form, MBL, which is something that seems not to progress to CLL. And one of the things that I think one of the most interesting things we can do with genetics is actually distinguish the people that end up having clinically relevant disease compared to the ones that don't. That's certainly the research goal in the CLL. Prostate cancer is another really good one. Many men are diagnosed with prostate cancer that are not clinically relevant. By precaution, we tend to overtreat those ones, and I can understand the rationale to do that. But if we can better distinguish the people that are going to develop clinically relevant disease based on their genetics, I think that's one of the most potentially important things we could do with genetics, to try and work out the ones that are going to do badly and the ones that we probably should, as clinicians, not treat them. I think that's a really interesting idea.
Any more question from the audience, please?
Thanks for a great talk. Do we have enough evidence from genome-wide association studies to include this data in genetic counseling?
Well, we have some good questions today. I think that's a bunch of clever folk. That depends on the cancer, and it also depends a little bit on the genetic nature of those diseases. It's a big debate in the community at the moment. Can we get to the point where we can identify sufficiently, with sufficient precision, those who are going to develop the disease or not? And it's one of the biggest research questions that we are dealing with as a community inside genetics. And it depends a little bit on the different types of diseases. For example, in prostate cancer, there is some good evidence to show. There's a beautiful paper from some colleagues here that were working here at the IARC for a while, but now in California. And they looked at, can we use polygenic risk scores to detect aggressive prostate cancer after we, particularly using the PSA measurements. And there, it looks like there's real potential to identify individuals that will are at higher risk of developing aggressive prostate cancer. So there, yeah, I think we do. To do that, they needed a genome-wide association study in the order of 200,000 men with prostate cancer. When I look at lung cancer, we're not there yet. So we probably do need that type of sample size. And that being said, it also depends a little bit on the geneticness of the disease, the heritability, because CLL, chronic the form of lymphoma, there, even though we have much more sample size, the precision at which we can distinguish a risk is already pretty good, because that is a much more heritable trait. Okay, and there we only have in the order of 7,000 cases where inside our GWAS. We're expanding that at the moment. But there's the other side to that. How do we do it? And as quite rightly the question came up with CLL, many CLLs never end up being clinically relevant. I can identify that you're at risk of it, but I can't tell you if you're going to be sick. So then we have another ethical, and even worse in those ones, that's incurable disease. So I'm telling someone, you're at risk of this horrible disease, this manageable disease in this instance, but you're going to be at risk. And so there's a lot of morbidity associated with that particular thing. I'm telling people they're going to be stressed, they're stressed for themselves, for their children. So there's an ethical dilemma of, should we do it? And even worse, if you can't treat it, do you want to know? So it's a big debate at the moment. How do we best use that knowledge, and what's the framework that we want to be able to do and put that genetic knowledge into practice? I'm sure there is potential for it, but we just need to be very carefully that we manage in all screening mechanisms the benefits and the harm. To finish off, maybe I've got one slide, and I'll show you something about how we do that in lung cancer. So lung cancer, there is a screening mechanism out there. Okay, I'm just going to explain what this is. This is lovely work that my postdoc Ricardo has been doing. There's a way you can screen people that decreases the mortality related to lung cancer. And it's low-dose CT screening. So if we can identify high-risk individuals, and that's usually done through smoking questionnaires, identify that you've got a good, strong smoking history of lung cancer, you're at high risk. If we use low-dose CT scans, we can decrease the mortality associated with lung cancer by 20%. There's a risk. I hope you just didn't cough because you're smoking, but there is a risk associated with that practice because there's some morbidity associated with it, but we can do it. Now, what this graph here is showing is that at what age do you achieve the risk, because it's a threshold. It's a threshold of risk. If you achieve a risk, an absolute risk of developing lung cancer in the UK, it's 1.5%. In the US, it's 1.3%. You are recommended to low-dose CT screening. It's a cost-effectiveness thing. And what we're doing here is plotting the different genetic risks. The red line is the genetic risk, the low genetic risk individuals. Sorry, beg your pardon, I get that wrong. The red line is the high genetic risk. The green line is the low genetic risk. And what we see is the age at which you achieve that risk is very different between those two individuals. So we still use the questionnaire, do you smoke? But what we use is the genetics to recommend at what age you should be screening. And so this is where we're trying to do this as the community, is trying to come up with the best ways to incorporate this type of genetic effects in our clinical practice. I should probably stop.
Thanks. Thank you.